actually presented in this conference in different sessions is the translational aspect of gene therapy, starting for experimental and preclinical research with basic science on gene regulation and system for gene transfer, animal models, and if they're successful, then we have to move to more complicated cells production at the scale of a human being of gene therapy products abiding to GMP and GLP guidelines and rules at the international level. And here you can move to clinical trials which have to abide to the EC directive on GCP, which is of utmost importance. So the administration route in vivo or ex vivo is not the same. Here is a direct administration of a vector in a bottle. And ex vivo is supposed to have cells taken out from the body, grown in vitro, transduced, and then put back in the body of the patient. So these are steps that are different, and in terms of risks that you can encounter with direct administration and with gene-manipulated cells ex vivo, it's not paralleling, and of course each is a different story. We also have a broad range of technology now into place with the AAV platform, the Retrovirus platform, including the gamma retrovirus and now the lentivectors, which are really showing success in the clinical area. Cell therapy, adenovirus, and non-viral methods, including transposons, and I believe there's a very interesting talk and session about these vectors soon in this conference. So, gene transfer systems like vectors can be used according to the type of cell you want to transduce. Of course, you will have to use a different one. And if we draw the ideal vector profile, we in fact are drawing the ideal option in terms of ethical choice, because if we get that good a vector, which will be convenient and reproducible to produce, which will be directly injected in vivo and targeting only the cells of interest you want to target and integrate into a site-specific manner with a transcription unit that can be regulatable and which induces no immune response. Basically, you will have solved all the ethical tensions that we have now dealing with gene therapy vectors, which are imperfect. So currently what we have to do is trying to do in an imperfect world with what we have and try not to harm patients with these tools. So if we consider altogether the associated risk to gene therapy, we have to consider that there might be carrier toxicity like the gene transfer vector, immunotoxicology and immunotoxicity due to the vector, but also to the transgene, perhaps. We also have, in some instances where there is out of target expression in cells that are not supposed to express a transgene product, we can also face phenotoxicity, genotoxicity of the vector with insertional mutagenesis up front, transgene silencing, meaning that the expression is shut off, vertical transmission and horizontal transmission. Vertical transmission by guidelines according to the EC regulation is strictly forbidden. This is something that you cannot accept in a clinical trial. So basically, if the vector is shown to go into the sperm or the ovocytes, it cannot be elected for a clinical trial, even if it's life-threatening disease. This is the way the regulation is drawn, and we can think of changing it, but this is the way it is. So you can see through these pictures that the scope of risks that can be envisioned with the new compounds that we're dealing with is massive. So, for example, we know that the adenovirus vectors, initially seen as a high, potentially versatile vector, in fact now is, since the immunogenicity is prominent, is mostly seen and envisioned as a vaccine. If some of you are interested in the slides, I have given them to the organizers of the meeting. So technology-related list with AAV, we know that it's a non-pathogenic virus, as we've been told. It was first thought as a non-integrating vector, but it might require re-evaluation, and there is side integration of AAV for sure, that perhaps will need consideration in the future. The main drawback, current drawback of AAV, of course, is immunogenicity. So you know that the first trial that developed in hemophilia was stopped because of this immunogenicity, and studies in non-human primates were further performed, and recently a successful trial was performed with improved technology. So in upgrading the level of technology and changing it, the investigators, I'm sorry this is not going through, doesn't show. Oh yes it does, sorry, I need to click another time. So the investigators decided to move to the AAV serotype 8 in order to go around pre-immunization in human and try to go around this immunotoxicology that we just mentioned. Again, another feature of this vector is to be SC-AAV vectors with specific expression cassette for the liver. And so the potency is ameliorated, and lower doses of vectors could be used. And the third feature of this trial also was to be able to deliver the vector into the peripheral vein due to the use of this serotype and to the targeting of the expression into the liver. Altogether, this translated into at medium and higher doses of serum level of hemophilia 9 factor, anti-hemophilia 9 factor, to levels that could bring some correction. And as you see, the cell-mediated immune response against AAV capsid that was observed in the Pennsylvania trial run by Kathy High was milder and was not sustained in time, as you will see even at the higher doses. So now more patients are being enrolled in this clinical trial, and it's doing better. Coming now to integrating vectors, as you know very well, perhaps, retrovirus vectors and mostly gamma retrovirus vectors have brought the best, have both the best and the worst. In the late 90s, the best was that SCID patients, babies born with immunodeficiency, inherited immunodeficiency, were going out of their bubbles and could eventually go to school. This is the same with ADA and does stand as a reference treatment at present, which might go to the stage of the marketing authorization at the level of the medicine agency, and also CGD, chronic granulomatosis and epidermolysis bullosa. But very soon after this first successes, the worst happened under the form of leukemias as a direct consequence. I mean, the connection between the gene therapy intervention and the fact that leukemias were happening was directly established. So you think that you are treating a disease, but you are bringing leukemia to patients, and half of them were getting leukemias with time. Still, what does the risk-benefit ratio tell us? This is something to think about in ethical terms, because if you think of these patients who had no treatment option other than gene therapy, then even half of them surviving might be better than none. But this is kind of extreme, and we have to think of how we can ameliorate this tension. And hopefully enough, recent advances with lenti-vectors-based trial show that, for example, in Wiskott-Aldrich syndrome, where the leukemias were happening with the gamma retroviruses, they do not happen with lenti-vectors. So this is good news, and again links the ethical tensions to the level of, to how the technology progresses and is improved. So what we learned from these leukemias in patients is that insertional mutagenesis in a gene like LMO2 can be cis-activating the LMO2 gene by retroviral insertion. This is for a fact. So the European Medicine Agency retained multiple factors involved in this leukemogenesis after retroviral mediated gene transfer, and some were vector related, and as I have just explained, what might happen with gamma retroviruses with viral enhancers and promoters might not happen, hopefully, with lentivectors. Related to transduction protocols, the fact that you are overstimulating the cells with a lot of cytokines might be, of course, a potential explanation for some of part of the inconveniences. The number and expansion of transduced cells, the integration sites, of course, in open chromatin, the nature and expression level of the transgene might also be, of course, of concern. The genetic host factors and what we see in mice does corroborate this fact, because as Ginio Montini showed yesterday in the satellite meeting on the genotoxicity retreat, the data that we have together show that if we have cancer-prone mice, they are able to develop tumors where mice that are not cancer-prone will not develop. So there is a way to sensitize, and some patients might be more vulnerable than others due to their genetic buildup. Acquired host factors might also come into the way, and of course, serendipity. So what are the ethical issues in clinical gene transfer research that I'd like to point out? So why? Why is there an expectation that gene therapy intervention should be final and at once? You know, there are many medicines that you take every day in a chronic disease, and that patients accept to take for a long time. But with gene therapy, obviously, what the expectation is for final cure immediately. So let's take some time before we can achieve that and consider whether gene therapy might be a good choice or are there safer alternative treatments. When? What extent of animal experimentation is needed to start thinking to move to the first clinical use in human, what we call FIH. So special consideration should be given to the stage, again when? To the stage of the disease where gene therapy is hoped to be effective. I always like to mention the situation of the Duchenne. You might be aware that at the age of 1, a Duchenne kid has already lost 80% of his capacity to generate stem cells in the muscle. So if you don't go early enough in the course of the disease to try to genetically modify those cells that are able to be stem cells and be corrected and generate daughter cells for a while, then you lose the point, and there's no way gene therapy is going to be efficient in this context. So the case of orphan diseases without alternative treatment is, of course, very important. How does point on the design and safety of clinical trials and the ethical impact of improved technologies. I hope I have shown you how much technology can alleviate some ethical tensions and perception should the technology prove safer than the fact that you're moving forward is, of course, becoming obvious, and less toxicity means more opportunity. So this is evolving knowledge. So the focus is really on this pivotal question, when to go clinical. So we need to draw a line between the requirement for between investigational new drug and what it entails, and the fact that anyway there will be basic research run in parallel. There's no way we can escape the clinical stage, and there's no efficacious treatment without drawback. Still, we know that we are making progress, technology progress, like I just mentioned with lentivectors and, for example, with AAV in hemophilia. So when should we start thinking of treating a patient when we know that we need to ask how a vector system or a rational will work in patients, and at the same time there might be more terrific options in 5, 6, or 10 years to come. And what we currently know may evolve. So this is the real ethical tension is here. When do you start to begin is the real question. So moving from bench to the bedside, I'd like to quote here Nancy King, who's been working with the Kidney Network of Excellence. She's from the United States, and she has been working, we've been working together in a terrific manner, exchanging very critically and in a very coherent and productive way. So she's been lecturing at many ESGCT conferences where Kidney Network of Excellence wanted to share her wonderful experience and the way she is teaching, in fact, ethics with this audience. So this slide comes from Nancy, and what we need to do before we go to the bedside is to ask 3 questions. Has enough preclinical information been collected so that the only reasonable way to learn more is to start a clinical trial? This is extremely important. If you have exhausted every possible option that you can test, in vitro or in animals, then maybe you can think of going to people and to patient subject. Second, has enough been done to reduce the risk of harms to human and to maximize the likelihood that the intervention might ultimately show benefit or at least no loss of chance in regard to the reference treatments or other options that might be possible. And has the point of irreducible uncertainty been reached? I mean, irreducible uncertainty is something very important as a concept, because then it might be fair to subject to ask them to become involved in the research if we think that the uncertainty has been reduced to the maximum we can with the tools that we have and the knowledge that we have at present. But of course we know that this irreducible uncertainty at one time point will be different 5 years later, when you will have learned more from more clinical trials and the fact that the rationals and the technology will have been tested in humans. So balancing harms and benefit is something that is built up into consideration of clinical trials, and you find it in the Nuremberg Code, the Declaration of Helsinki, or Helsinki Declaration, and the Belmont Report. So basically, medical research involving human subjects may only be conducted if the importance of the objective outweighs the inherent risks and burdens to the research subject. So in clinical trials, the balance between the risk of harms and the benefit is always paramount. So how can you try to make your way for a decision-making into an imperfect world where you know that you might not be dealing with the best of the technology, that research is still ongoing, but still you want to try to test something in a clinical setting. So we went for a model, thinking for a model for designing and planning early phase clinical gene therapy research, and propose a stratification scheme built up on these few concepts. First is subject selection. Which first subject can be sufficiently protected from harm is one question. And second question, from whom, from which kind of subject may the most be learned at the least risk to them? So sometimes you can think that very sick subject may be considered because they have nothing to lose, quote. But they may be at greater risk of harm, but they may value chances of benefit highly. Well, in this context, it is not acceptable to take people because they are very sick if there's nothing to learn from the situation. Basically, if you like to consider very sick people to enter into a clinical trial, then perhaps there will be no benefit for them and it's too late. But then generalizable knowledge has to be accumulated, and at least you need to think that they will provide data that will reinforce the level of knowledge that we have at this point. Again, in this subject, the fact that the disease has progressed, the fact that they might have many therapeutic interventions in the past might be hard to disentangle with the new treatment that you are going to provide them with. Healthier subjects sometimes are so healthy that if you give them a treatment, it's too early in the course of the disease to hope to see some benefit appear and to see anything that would be readable at the later phase trial. So which first subject are most likely to benefit will depend on the condition and on the technology that you are dealing with, and it's extremely important to think of the subject selection. Then is the question of how severe is the disease. If there's in instances where you are dealing with a life-threatening condition without any therapeutic option, moving to gene therapy might be justifiable because there will be no loss of chance, first, to benefit from existing treatments. So if you don't have any other treatment, or it's not expected to be as good as gene therapy, then it might be a point. It's unlikely to leave patients worse off or to shorten their survival. That's also very important. It's not, again, because patients are very sick that you are authorized to shorten their lifespan. And again, this very important notion that I just brought up, increased generalizable knowledge is clearly a duty that we have and a due to society. If we perform trial, we have to bring up the knowledge that we can take from it. So, as I said, safety is first. There's a few hints for benefit, and this is not a primary objective, but you can learn, even if there's no direct benefit, how to proceed further and to go to achieve cure in the next step. So we have to satisfy both the imperative to protect subjects and the need to move the research forward. As I say, this is the tension, but this is our duty to proceed further, but also not to harm the patients. So I think I already told you about when in the disease course to undertake gene transfer therapy. We have covered it before, so we can move to the model stratification scheme, which in fact is not specific to gene therapy and could be used, of course, for stem cells, as I will show. But it's a combination of the technology, the disease characteristics, the risk of harm and potential benefits, and this requirement of justification we'll see will be articulated in the consent form. So this is a kind of, as you see it, it might be complicated, but I will try to drive you shortly in the 3 minutes, and then we'll be close to the end of my talk, to this stratification scheme towards performing first-in-human trials in patients. So if you start with the disease stage, at the end stage you have the sickest person. As I told you, in this person who's already very sick, you cannot hope for direct benefit. The goal of the study is safety. So there is no direct benefit, and the performance of a clinical trial will be to improve generalizable knowledge, as I mentioned. If the therapeutic window is closed, like a kid with Duchenne who has no more stem cells in his muscle left, there will be no cure and no direct benefit. And no risk-benefit can be assessed. But you can look for safety, and if the safety record is okay, then you will be able to perform next, first in human trials, in less sick patients. So this is what you can learn from the sickest patients. When there's a life-threatening disease without established treatment, then the goal is also safety. And at first, there's no direct benefit to expect, but if the therapeutic window is still open, then you may also collect some hint from direct benefit as a secondary objective. And then through these situations here, you can collect knowledge which will bring you more options in the future for life-threatening diseases with alternative treatment, because you will be able to make a balance between the risk and benefit and see which treatment will be first. Will you start with gene therapy, or will you start with the alternative treatment? Let's take the example of SCID, or BEST, the ADH-SCID. You know that in the case of ADH-SCID, you now have an established treatment with gene therapy that can be very safe. If you compare it to bone marrow transplantation, then if you go gene therapy first, and if it doesn't work, you can still perform marrow transplantation. Whereas if you go marrow transplantation first, you will never be able to perform gene therapy, because you won't have any target cells left to try gene therapy in these patients. So the rationale is to try always to make the balance between risk and benefit, and if it has been shown safe enough in this situation, then you can begin to ask the questions of gene therapy versus alternative treatment. Of course, the Holy Grail is here pictured. If you have the LCS patient, then the goal will be efficacy. Direct benefit is clearly the aim. You can work there with technology that has been shown safe, of course, and with which all preclinical model will be okayed, and cure is possible, and risk-benefit can be evaluated, and the ultimate goal is treatment. But before you can fulfill this Holy Grail, you perhaps have to go through this scheme first, and I'll let you think more about it. This has been published back into 2008 in Molecular Therapy, and we are preparing another paper to explain better the thinking and the rationale of this stratification scheme. To finish with is the informed consent, and this last but not least. As I told you, this is a contract between the principal investigator, the cure team, and the patient. So it has to explain why it is necessary to learn from humans and fair to ask them to participate. We have to inform patients on the FIH primary endpoint, and of course, again, we have to put forward that it is to establish safety first. We have to explain the risk of harm and benefit balance, describe the risks of harm and their minimization as far as we know. But of course, we have to mention also that there is uncertainty, and there are still things that we don't know, and the unknown might be coming soon and show in the way during this new clinical trial. So we have to explain how and why research is not treatment, and an intervention as a first-in-human administration should be considered as a research and not a therapy. You cannot say to a patient, we are giving you this treatment, because we don't know really if it's going to treat them. It's a product that we are giving them, and we are looking for the treatment ultimately. So we have also to emphasize the research partnership and avoid inducing the therapeutic misconception and false hope. Meaning that in many cases, as I've shown you, what we will learn from a first administration is human is generalizable knowledge that will tell us how to do next in perhaps patients that will be healthier ones. So there is no way we can promise a patient that because he's entering into a clinical trial, he will have some expectation that his condition will improve. But as a secondary objective, if we think that there's a potential that some positive aspect and direct benefits might show in the way, then you should be able to explain them, but not to provide them as the goal and the justification of them entering into a clinical trial. So I think the conclusions and recommendations are just summarizing what I just said, and I don't want to read them loud while you might be starving. So if you're interested, you can ask for the PDF on these slides to the organizing at the reception desk, and they will forward them to you. What I'd like to finish with is how ethical consideration in the field of gene transfer, which can be considered now as a very mature one in the regards of the ethical consideration and the fact that clinical trials have been performed with few expectations for progress and benefit in early phases, but now with clinical benefit entering into space, but how the field of regenerative medicines can be concerned. The ethical issues, there are conflicts of interest, and with many companies producing many compounds, it might be difficult to develop. You've shown us how many companies can be involved, and at the end you may want to mix 2 or 3 different things together to make it work better, but maybe not because of conflicts of interest. So this is a key issue in the field of regenerative medicine, as we feel is the current situation. Technological challenges and uncertainty do apply to the field of stem cells and regenerative medicine for sure as well. This is shared. Experimental transplantation procedures with or without genetic modification are really similar issues that we have covered when considering gene therapy previously. The use of embryonic stem cells I will leave apart, but the use of induced pluripotent stem cells with or without genetic modification do interrogate us and bring us back to the field of gene transfer and genetically modified cells. As you know, pluripotent embryonic stem cells can generate every cell in the body of a human being. But there are 2 ways now to try to think of developing a regenerative medicine approach, either through the ES cells, and you know that in some European countries still there is no way they can be used, whereas induced pluripotent stem cells from adult cells, autologous adult cells, can now be generated and able to generate ectoderm, mesoderm, endoderm, but not germ cells. Well, this now is a question mark, actually. So in a paper that we published earlier this year in Current Gene Therapy, we showed what are the different delivery methods that can be used in order to generate iPS cells. And you can see here that basically we're covering the whole field of gene therapy vectors. And despite the fact that gene therapy is getting more mature, what we are currently facing is a situation where people working with iPS might use vectors that are less refined in thinking and in technology than the gene therapy community is using. So I leave you to your reflection to have more interaction between the 2. So the advantages of iPS cells is that they are ethically acceptable because it's an autologous adult cell, and it's not an embryo that is being distracted in order to generate stem cells. The simplicity of the protocol is, of course, attractive, you can generate many IPS for reflecting many disease models in human. And with autologous cells, there is no histocompatibility issues when thinking of transplantation. And unlimited growth is obviously the case when you have fully reprogrammed iPS cells. The inconveniences which balance these advantages is the low efficiency, which is currently improving, by the way, the ectopic expression of oncogenes, and the fact that sometimes the sequences are not deleted and may integrate or left integrated. This is why we think that developing exchange cassettes is really the way to go. The risk for insertional mutagenesis, like with integration, as we mentioned with the gene therapy aspect, and the safety taking us to consider DNA integrity. Genetic instability, genetic aberrations, and epigenetic changes have been observed and described with iPS. And here also is from a review of De Vos and collaborators in current gene therapy, the dark side of pluripotency, and the causes of genome alterations that one can observe in iPS cells. It seems like not all of these genetic alterations are caused by the reprogramming step itself. Sometimes the mutations are pre-existing. Sometimes there is in fact insertional mutagenesis, and sometimes there is also a result of the culture growth, which is still a question mark. So you see that when we are dealing with new technology, there are new questions coming up, and part of the questions that have been asked in a field like gene therapy, gene transfer, might also apply to another field like stem cells and regenerative medicine. This is why I was very happy to have your talk before mine, because this was obviously a regenerative medicine talk. So the credit to this work I mentioned goes first to Nancy King from Wake Forest University, and the Clinigene Network of Excellence, Émilie Bayard, my collaborator. Some pictures from the video that you can have online on the Clinigene website explaining the basic principle of gene therapy. This is a wonderful video, and you can go to the Clinigene website, click on the video, and have 15 minutes of pure science and jubilation. Federico gave me some slides, just as Mark Bichonsky on the regenerative medicine, and John De Vos for the publication in current gene therapy. With this, I'd like to leave you with the notion that the Clinigene Network of Excellence finished is, of course, with this, the publication of this clinic book, in which all these aspects and the ethics are also being covered, together with the regulation of gene therapy in Europe, which is not a piece of cake. And I'm sorry that I could not project this presentation from my own computer, because you will have enjoyed this, the Buena Vista Social Club music from La Habana. And on the wall of La Habana, they are writing "Caemos en los sueños." I believe this is Spanish, and this is the best place to quote that hope lies in dreams. And this also is the thing that you find on the wall of the Salk Institute in La Jolla, and that Jonas Salk took as a motto, and is quoted in the clinic book, actually. So I leave you with "Caemos en los sueños." Without music, but with a lot of hope that it will help you down your way to developing clinical trials. Thank you for your attention. We may have time for 2 burning questions, if you have one. Yes, please.
Yes, a very impressive your presentation. 2 questions. If are some approved and validate clinical study using gene therapy, as was presented before as a first successful clinical apply, and In your opinion, if a hospital wants to apply directly in the 2 or 3rd clinical study, it's possible? First question. And if not, it's possible for European Gene Society to offer help and support to overpass this obstacle. Thanks.
Wow. Well, to come with the first obvious question, the second one is simpler. Well, that was clearly one of the goals of the Clinigene Network of Excellence, to be helping people going through with a project. But unfortunately, or fortunately enough for me and my timetable, because I wouldn't sleep at night for many years, so now I enjoy resting a little bit. And so to tell you that this project is terminated. So what I would advise you to do if you have a project and need some assistance is to go to the European Medicine Agency, where they have a protocol assistance, and they will basically gather scientists for you. I'm not sure that the European Society has put together the necessary group or committee to do that. Maybe that should be a suggestion that you send to the society. But at present, you know, the focus of the society is to organize a very good meeting every year. I don't think that they can advise people with project, but if your project is mature enough, the European Medicine Agency has something that is called protocol assistance. So you can go to them. The second thing is to make a protocol available to a hospital who has a potential for patients and recruitment in this area. Would work if there is, I think, a private sponsor and industrial sponsor to this trial. And at present, we are starting with clinical trials in gene therapy, and many of them are academia-driven. So it's difficult to ask colleagues who are looking for funding for their own trials with 3, 4 patients to come to your facility and to bring their protocols unless you will find some funding for that. I think that I was trying to show to you the tensions at the ethical level. The next thing is, if the ethical tensions have been relieved because efficacy is showing and safety is showing along the way, then the next step is to make this treatment, as I said, I mentioned that under justice. How can you make this available to a lot of patients who might need it? Was that your question? Ah,
The question was very practical. I'm a hospital, I have funds, I have passion, and I want to apply, validate, and approve gene therapy study. In your opinion, I can or not?
So you mean there is a group who has shown efficacy?
Yes, first presentation was presented as the first successful clinical study until the final. This means the 3rd stage.
But again, I think this is the thing that will retain from the development will be conflict of interest. It's not if you don't have funding issues, if you don't have the will to share things, will be the limitation. So if it's academia, it's likely that people are going to share with you and bridge with you, especially if you have your own funding. Because from the ethical or regulatory standpoint, there is no issue. You have a file, it's established, and you can go Europe-wide with this file. It will work.
Then it's not a problem from ethical issue.
No.
Okay, thank you.
No, certainly not. Well, if there are no other questions, I admire this audience for staying so late and listening to this talk. If you have questions, you can direct them to me by email. Thank you for being here and for listening. Thank you so much.